Let's discuss on cancer biochemistry. Cancers refers to any normal growth of tissues. It may be benign or malignant. It may also called as neoplasm. And what is the biochemical importance? It is the second most common cause of death worldwide. Humans of all the ages are affected and wide variety of organs are affected. Then cancer, the term is usually associated with malignant tumors. So it may be defined as a malignant neoplasm as well. Cancer is a disease in which some of the body's cell grow uncontrollably and is spread to other parts of the body. All cancers are multifactorial and they include the genetic, hormonal, metabolic, physical, chemical, and environmental factors. The most common cancers are spontaneous. The cancer originates usually from one aberrant cell which goes on multiply and produce a tumor mass. As the age advances, the number of mutations accumulate and hence the statistical probability of incidences of cancer among the adults and the aged people is more. So any substance which increases the rate of mutation can also enhance the rate of incidences of cancer. Therefore, all the carcinogens are known as mutagens. For example, the X-ray, gamma rays, and ultraviolet rays, these are all mutagens. Some human cancers are caused by chemical. These may be introduced into the body. Etiology can be from occupation, like inhalation of aniline or asbestos. It can be the diet, aflatoxin, it being derived from fungus, lifestyle, smoking may be another reason. Then chemical carcinogens act cumulatively. Then tobacco, food additives, coloring agent, aflatoxins are the common carcinogen in our environment. Now, what are the anti-mutagens? Anti-mutagens are those substances which interfere with the tumor promotion. So there are a lot of vitamins involved in this process, like vitamin A, vitamin E, and vitamin C. They prevent the production of new cancers. Then curcumin or the turmeric or haldi we use in our house is known to prevent mutation. Fiber content of the diet beneficial to decrease the colon cancers. Then flavonoids possess anti-mutagenic properties. Beside the phenolic compounds found in fruits like grapes, strawberries, walnut, etc. are found to be anti-mutagenic. Now here have a look onto the oncogenic viruses because these are also another etiological factor causing the carcinogenesis. So how do they integrate to the host DNA? So first the viral DNA takes it entry into the cell. Now viral DNA attaches the host DNA. This is the sort of attachment. Then strand transfer happens. It incorporates itself into the host genome. Further, the nicks are being sealed enzymatically. Then finally, the virus DNA is now integrated. So this is another important reason causing the mutations and therefore carcinogenesis. Then oncogenic viruses causing the cancer among the various organisms. So there are three categories. One is the Papova virus group. Second is the retrovirus type C and retrovirus type B. So retroviruses are basically the RNA viruses. They have got the reverse transcriptase enzyme capable of producing the DNA. Then let's see, this is the SV40. Its genetic material is DNA. It causes the sarcoma in the mouse. Then papilloma, that is also a DNA virus and it causes papilloma in the rabbits. Then Marek, it's the chicken, it causes the lymphoma of chickens. Then retrovirus C type. So there are two categories. One is the gross retrovirus. Second is the ROS. The gross is the RNA virus and it causes the leukemia in the mouse. Then Rouse causes the sarcoma among the avians. Then retrovirus type B, so Bittner virus, it causes the mammary tumors among the mouse. So this is how the various diseased production has been observed among all these animals. Besides that, there are various human oncogenic viruses being reported. So like Epstein-Barr virus, it is abbreviated as EBV. It causes the Burkitt's lymphoma and nasopharyngeal carcinoma. Then human papilloma virus, that is the HPV, causes the uterine cervical carcinoma. Then hepatitis B virus or HBV causes the hepatoma. So here have a take a look onto the how this human papilloma virus causes the cancer in the cervical, uterine cervical region. So this is the infection caused by the HPV. Now it enters the tissues in the uterine cervical epithelium, causing no, it here replicates, propagates its growth, but these epithelial cells they heal up very fast. More than 90% of the epithelials they are healed within 2 years because of the strong immunological response of the body. So normally the cancer doesn't manifest, but among 0.8% or to 1% of the people it has been observed. Like after 10 to 30 years, if it persists in these sites, this viral genome, it inserts itself into the host DNA. And further, this is how it is causing the cancer. And it is breaking this, you can see the breaking the basal cells, basal layer, and now it is infecting to the other tissues in the vicinity. So this is the how the this human oncogenic virus causes the uterine cancer. Then oncogenes are the genes capable of causing the cancers. So in raw sarcoma virus, the full virus produces sarcoma in the avians, but a strain of the virus which is deficient in particular gene could not cause the disease. So the oncogenes present in the normal cells are also called the protooncogenes. So products of many protooncogenes are polypeptide growth factors. For example, the SIS gene, cis gene, produces the platelet-derived growth factors, and the factor is required for normal wound healing. Some of the product act as a receptor for growth factors, like RB B produces the receptor for epithelial growth factor. Here, take a look onto the table. It's the table concerning with various oncogenes like ABL genes, ERB, BV gene, MX gene, cis gene, RAS gene. So what are the viruses like Abelson leukemia virus in mouse? It contains this oncogene. The oncogenic product is the tyrosine kinase, and the subcellular localization of this oncogenic product is the plasma membrane. So uncontrolled production of these gene product due to the activation of oncogene will be the reason for carcinogenesis. Similarly, the erythroblastose is a virus in chicken, and the receptors for it produces the receptors for epidermal growth factor EGFR. Then the site is membrane. Then similarly the MEC, it is the myelocytome A virus in chickens. It is the DNA bonding protein, and the site is nucleus. Cis is present in the membrane, and it is present in the simian sarcoma virus in monkeys, and platelet-derived growth factor is the product. Then RAS virus, it is present in the cytoplasm, and in the rat sarcoma it has been present, and the product is GTPase. Then oncosuppressor gene. These are the genes which normally protect the individual from developing a cancer. When a gene is deleted or mutated, the cancer results. So oncosuppressor genes are truly beneficial to us. It's a balance caused by the various anti-oncogenic vitamins and substances and the oncosuppressor gene, and they are inhibiting the generation or no sustenance of cancer. Then P53 is a gene encodes for phosphoprotein. It is a very popular oncosuppressor gene. We'll discuss on this. And this is the how the because a lot of proto-oncogenes are present in our body, but we don't suffer from the cancer because these proto-oncogenes, these are the inactive genes. So they are not capable of causing the cancer until unless they are getting activated. So what can be the causes? They lead to the activation of these proto-oncogenes. Besides, the oncosuppressor gene all time work in order to protect the individual from developing cancer. So we will see how the proto-oncogenes are activated. What are the ways they converts up into the active oncogenes? So one is through deletion phenomena. And base pair deletion will lead to certain changes in the nucleotide framework of the DNA, and that will be the reason for mutations, and that will lead to the truncated protein with a loss of function. Similarly, the translocation of the chromosome, that can be the another reason for causing mutations in the genome, and that leads to this translocation of another protein coding region and fusion to the present position. That will lead to like kind of a lot of altered, functionally altered proteins. So synthesis of fusion protein with loss of regulation. Then another way can be the insertion of retroviral gene. So retroviral insertion and then activation, that will lead to the increased synthesis of abnormal increased synthesis of proteins, altered protein. Then regulatory sequences, they translocated from some other site. So there can be three major reasons: deletion, insertion, and the chromosomal translocation. They may be responsible for activation of these proto-oncogenes. And the resultant is the functionally altered protein. Then here is a list of a number of oncosuppressor genes. One is the retinoblastoma gene. It is abbreviated as RB. Then Wilms tumor, abbreviated as WT. Then familial adenomatous polyposis, FAP. Then deleted in colon cancer, DCC. Then gene for protein 53, that is P53. Then familial breast cancer oncosuppressor gene is the BRCA gene. So these are the various oncosuppressor genes. Now let us take a look on various characteristic of cancer cell. Cancer cells are characterized by certain main properties, like they proliferate rapidly, they slow, diminish growth, and so a malignant transformation. So when a normal cell has acquired the malignant character, it is said to be transformed. So normal cell is generally monolayer, but because of uncontrolled propagation and proliferation, they cause multi-layered appearance. Then contact inhibition, it is characteristic of the normal cell, which is absent among the malignant cells or the cancer cell. So if a cut is made in the skin, the cell from both the sides starts to multiply, but this multiplication is stopped when the cell come in contact. So this is the phenomena happens with the normal cell. Cancer cell have lost this capability. Then metabolic alteration in the cancer cell. The cancer cell prefer to thrive on minimal enzymes. And Warburg hypothesis says the cancer cell favor only the glycolysis, even in the presence of huge supply of oxygen. They don't go for oxidative phosphorylation, so they are dependent on glycolysis for their energy needs. So why cancer cells are immortal? One reason is that cancer cells have increased in persistence activity of the telomerase. And this telomerase enzyme, it maintains the length of telomere, and that's how There is never ending short of the chromosome. No shortening of the chromosome. It remains immortal. So cell keeps going on with the division. Then apoptosis. Apoptosis is a programmed cell death caused by CKDs and cyclins. Uh, then it is the example of apoptosis mediating genes, and they are called C4s and P53. That is also a tumor suppressor gene. So they are meant for this apoptosis phenomena. And so they in turn are oncosuppressor gene, and by the same token, the apoptosis protecting genes are the cancer causing genes like BCL2, like B cells, leukemia, or the lymphoma type 2 and other oncogenes. So this is how the apoptosis is. The apoptosis is not happens in the cancerous cell. Then oncofetal antigens. During the fetal life, a particular gene is active and the product, that is a protein, is therefore produced in the cell. During the differentiation process, this gene is suppressed and therefore the protein is not present in the adult cell. However, along with the malignant transformation, the de-differentiation occurs and the gene is de-repressed and the protein again is available in the cell. So these are the fetal proteins which are present in cases of the patient suffering from cancer, like alpha-fetoprotein, carcinoembryonic antigen. So we will see how it happens. So in this diagram, this is antigen. This is the normal adult cell. So this antigen is not expressed. The gene is repressed. Gene for fetal proteins production is repressed here. But if the cell is malignant, so what is happened? There is gene is de-repressed, and then again there is an expression of this embryonal antigen. So these are called tumor antigen, act as a marker for a detection of any disease, cancer growth. Then here in the embryonic cell, in the normal embryonic cell, the functional gene, and it is normally producing this embryonic antigen. So there is an expression of this embryonic antigen like AFP, CEA, but which is not happening in the adult because of gene. The concerned gene is repressed. But among the tumor and cells, this gene is de-repressed, and again it starts expressing this embryonic antigen. Now let's talk on tumor markers. What are the tumor markers? These are actually certain markers overexpressed by the malignant cells. And they are also they are usually the normal cellular constituent and that are present at normal or very low level in the blood of the healthy person. But their concentration alarmingly high among the tumor cells. They in the blood they are detectable. So they are also called the tumor index substances, and they are the factors released from the tumor cells and which could be detected in the blood and various other body fluid. Therefore, they indicate the presence of tumor in the body. So this is the oncofetal products like which are acting as a marker. So there are AFP, alpha-fetoprotein. Its level is alarmingly high in the hepatoma and germ cell cancer. So it is marker for these hepatic cancer and germ cell cancer. Then carcinoembryonic antigen. Its level is raised in the colorectal, gastrointestinal, and lung cancer. Then there are various carbohydrate antigens like CA125 and tissue antigens and tissue polypeptide antigen. So in ovarian cancer, the CA125 has been increased. Then this tissue antigen and tissue polypeptide antigen they have been found in journal Cancer. Then various other enzymes which acting as a tumor marker like alkaline phosphatase. So in bone cancers, it is alarmingly high. Then placental type ALP, it is indicator of lung and seminomas. Then prostatic cancers, prostatic acid phosphatase, that is PAP, PAP. that is indicator of prostate cancer. These are all enzymes actually. So there is a groupism of these tumor markers to which category they belong. So we are talking of enzymes, ALPA, then we have PAP, and then PSA, prostate specific antigen. This is not an enzyme. It is also been found raised in the prostate cancer. Then neuronal specific enolase. So nervous system tumors, it has been found to be raised. Here is a list of certain hormones. So like beta HCG, it is increased in choriocarcinoma. Then calcitonin, it is increased in medullary thyroid carcinoma. Then big ACTH, lung oat cell cancer. Then vasoactive intestinal polypeptide. Then epidomas. Then vinyl mandelic acid raised in pheochromocytoma. That is the adrenal carcinoma. Then hydroxyindole acetic acid raised in carcinoid syndrome. Then tissue catabolic products like hydroxyproline in the bone metastasis. Then serum protein like immunoglobulins. It is raised in multiple myeloma, macroglobulinemia. Then Ben Jones proteins raised in detectable in urine. Then in the multiple myeloma patient, then receptor tumor markers like estrogen receptor and progesterone receptors, carcinoma breast and carcinoma uterus. Then HER2 receptor, human estrogen receptors. So these are raised in carcinoma of the breast. And these are the certain monoclonal antibodies used as an anti-cancer agents. Rituximab, trastuzumab, then bevacizumab. What is these actually? These are targeting, these are the, tailor made antibodies. They have been designed in a manner. So they are targeting a specific, antigen over the tumors. So there you have seen a list of markers. They can be present on the surface of tumors, and also their secretions are present in the blood. So on being detected, a number of monoclonal antibodies have been designed which are working against these specific targets. So what is the benefit of these monoclonal antibodies? Like they are affecting or targeting only the cancerous cell, not the normal cells, because these antigenic products are not available onto the normal cells, or they are present in very, very low concentration. So that's how this is a therapeutic regimen. They act as an anti-cancer agent. So rituximab, its target is CD20 on B cells, used against the cancer NHL, chronic lymphatic lymphoma. Then B cell leukemia is there. Then a HER2 receptor, the trastuzumab is the monoclonal antibody for HER2 receptors, and used in against the breast cancer. Then bevacizumab AB, that is the for working against VEGF. Then it is work against the colorectal solid tumors of kidneys and breast. Similarly, the alemtuzumab, it works against CD52. It is working against the chronic lymphoid leukemia. And then cetuximab, KRAS, it is for colorectal head and neck. Then pentimum, that is for colorectal cancer. Then imatinib, that is for that is the target is a tyrosine kinase. So this is for chronic myeloid leukemia. Here we can see the methotrexate is a very a very well-known anti-cancer drug, which is a competitive inhibitor of this folic acid, structural similarity with that of folic acid, except for differences of amino group. and this methynyl group, which is carried over this nitrogen. So what can be the other line of treatment? Surgery and radiotherapy are the most effective to reduce the initial tumor load. And these are the prime modalities in the treatment of solid tumors. Then besides, we can go for chemotherapy in leukemias, lymphomas, choriocarcinoma, and other widely disseminated malignancies. So these are the various drugs, anti-cancer drugs: methotrexate, 6-mercaptopurine, 6-thioguanine, 6-fluorouracil, and then cyclophosphamide. It is an alkylating agent. Then mitomycin, it is an antibiotic. The baki rest are the competitive inhibitors, because this is methotrexate is folic acid analog, while 6-mercaptopurine is a purine analog, and 6-thioguanine is also a purine analog. So what is the mode of action? Competitively inhibits the dihydrofolate reductase, this methotrexate. Then second is the cis-mercaptopurine. It inhibits the conversion of inosine monophosphate to AMP. Then 6-thioguanine, it inhibits the synthesis of purine nucleotide. Then 5-fluorouracil, it inhibits the thymidylate synthetase. And this cyclophosphamide, it in a cross-linking of bases of DNA is being inhibited. Then mitomycin C, it cross-speeds are formed between the DNA and base pair. So this is how these are the mode of action of various anti-cancer drugs. Thank